like it's leveled out. Okay, we'll go ahead and get started. Hello, everybody. Good afternoon, good morning, and good evening to you, depending on where you may be calling in from around the world. I am Rob Burgess. I'm the Chief Business Officer for Biological, and it's my pleasure and honor to host the next installment in our immunodetection webinar Lock and Key series. We have two fantastic speakers here today from our new partner company, SignalChem Biotech, whom I will introduce in just a second. Before I do that, I would just like to mention that we are taking questions from the audience. So I highly recommend that you type your question into the chat box, and following the completion of the webinar today, we will scroll through the questions and try to get to as many of them verbally as possible. And also, please type into the chat box where you're from. We always like to know where people are calling in from around the world. And so without further ado, I'd like to introduce the two speakers here. The first is Dr. Adam Barlow. Adam received his bachelor's in chemistry from the University of British Columbia and later earned his doctorate in chemistry in 2017 from Simon Fraser University, both in Vancouver. During his doctoral studies, Dr. Barlow studied the photocatalytic DNA sequences, characterizing them from enzymological perspective. Following his graduate work, he took a position teaching biochemistry and other physical science subjects at Quest University in Squamish. Since joining SignalChem Biotech as the U.S. Central Account Manager, Adam has worked extensively with some of SignalChem's largest accounts and partners. Second, I would like to introduce to you Dr. Jesper Johansson. Dr. Johansson received his PhD in cell biology and genetics in 2017 from Simon Fraser University, also in Vancouver. And during his doctoral dissertation studies, Dr. Johansson worked on understanding the molecular mechanisms regulating the balance of endocytosis and exocytosis in yeast. He subsequently worked as a research postdoc and lab manager for a year at Simon Fraser before transitioning into a career as a university-level biology instructor. In his role there, he worked at the university for 5 years before recently joining SignalChem Biotech as an international account manager, applying his expertise to foster collaborations and drive scientific advancements on an international scale. So both speakers, we welcome you. We really appreciate your time today. And the title of the talk for Dr. Barlow and Dr. Johansson is Drug Discovery in Action, an Axel kinase inhibitor story. And with that, speakers, we will turn the screen over to you, and we look forward to your lecture.
All right, Rob, thanks so much for the great introduction. So you've already heard the title of our presentation today. Thanks for that great introduction. I'm Adam. I'm the account manager for the U.S. Central.
And I'm Jesper, and I will be taking the second part of today's webinar presentation. The first part will be done by Adam, so take it away.
Awesome. Yeah, Jesper and I were friends in grad school, but like Rob said, I'm more of a chemist and he's more of a molecular biologist, and you'll see that in our slides. So both Jesper and I are employed by SignalChem Biotech, which is a bioreagents and services company. But SignalChem Biotech is owned by SignalChem Life Sciences, which is a drug discovery company. But throughout its drug discovery process, it's been able to use the goods and services that are available to the public through SignalChem Biotech. We've recently entered a partnership with Biological. So right now you can purchase our SignalChem products through Biological. The future may hold other things for us that we're really excited about. But let's not talk about that. SignalChem Biotech is about 20 years old. We're about to celebrate our 20th birthday. We're located in the Pacific Northwest, but in Canada. And we're considered one of the world leaders in active kinase manufacturing and developing assays for them. Okay, so what are we going to talk about today? So first of all, I'm going to introduce the concept of a kinase. We're going to look at how to do kinase research, kinase assays, kinome profiling. I'm also going to talk about sort of the history of the kinase inhibitor drug market. And then I'm going to pass the mic over to Jesper, who's going to talk about a real example of drug development in the kinase inhibitor space, how SignalChem Life Sciences is taking SLC391 to the clinic, and how SignalChem Biotech can help you do the same thing. So what the heck is a kinase and where did it come from? Well, it's a coincidence, but it wasn't too far from here that the first kinase activity regulation was discovered. These two folks, Fisher and Krebs at the University of Washington, realized that there was an enzyme that released glucose in your muscles. It broke down muscle glycogen. And this enzyme existed in two forms. It existed in an inactive form and it existed in an active form. And they're the ones who figured out there was a kinase that converted the inactive form to the active form. And that this conversion, the kinase was listening to signals in the body. It was listening to things like insulin, the energy charge in the cell as a whole. And based on those features, it would decide whether it should activate this enzyme or not. And this is such an important concept that in 1992, they got the Nobel Prize for it. But this figure here that I'm going to use my pointer to point at, this is a really important figure that we're going to see over and over and over again in the presentation today. So a kinase is an enzyme that takes a substrate, takes ATP, also as a substrate, and phosphorylates that substrate, and ADP is released. Now, if that phosphate gets attached, if it comes from someplace else, sorry, not a kinase, okay? It's got to come from ATP to be classified as a kinase. This work was originally done in the 50s. And in the time from the 50s to now, it's been recognized that kinases are the key to understanding how cells control their fates. So kinases control lots of cellular functions. Something like 5% of the human genome is kinases. And the kinases act like couriers in a relay race. So an example of some kinases are that a lot of cell surface receptors are themselves kinases. And so they might activate another kinase, which then might activate one of its substrate kinases. You can also have multiple kinase networks converging and being integrated at one place. Another common paradigm that we're definitely going to see in today's talk is that cell surface receptor kinases, in order for them to function, they often have to dimerize. And then they also phosphorylate each other to become fully activated. The key here, though, is that the kinases have to activate each other in order for the signal transduction pathway to work properly. Now, this is so important to determining the fate of a cell, whether or not it grows or stays static, that all of these activities, the amount of kinase, the rate of kinase activity, all of that is very tightly regulated under a normal state. When we have a defect in kinase function, it's oftentimes possible for that defect to result in some kind of a disease, because these signaling pathways really require all of the different members to work properly for the message to be transmitted as intended. Now, a common kind of defect is that, for example, a cell surface receptor might become mutated so that instead of waiting for its ligand, which activates it, it can just be active all the time. And then it's constantly telling the cell, grow, grow, grow. You can see how that could cause a problem in the body. The idea of a kinase inhibitor is to block that signal and prevent that disease by modulating that signal. Okay, so what are these targets for kinase inhibitors, and how is it that we inhibit them? So let's take a look at the whole human kinome all at once. There's been decades of research into kinases that have produced beautiful figures like this one. And they're graphing all of the kinases that we know. And what we can see is that almost all of them are related to the same common ancestor. But even these weird exceptions here, all of them do the exact same thing. They take ATP and they take its last phosphate and they attach it to the substrate. So it's really ironic to think about it now, but for quite a while, this class of regulatory enzymes was considered to be undruggable because they thought, well, look, all of these enzymes bind the same substrate. They all bind ATP. How are we going to be able to find small molecules that distinguish between one kinase and another? But of course, we know now that kinase inhibitors are really common drugs. So at SignalChem Biotech, kinases are our bread and butter. We're proud of our kinases. So we have more than 90% of this human kinome in our inventory. And when there's some particular research use that needs a modification of a wild type, we have those as well. And when we find out that a kinase is mutated and that mutation leads to disease, we try our best to add that kinase to our catalog too, so that drug discovery scientists can use it in their programs. So how is it that kinase inhibitors work? So the first kinase inhibitor to be discovered was staurosporine. And the way that it works is that it enters the kinase and occupies the ATP binding site of the kinase. Without ATP, substrate can't get phosphorylated to the product, ADP can't get released. The problem with staurosporine, and the reason why it's not a drug, is that it's basically inhibits all kinases. So it's not really useful because it has so many side effects. But if what you're trying to do is do a kinase activity assay, and you want to use a good control that shows kinase inhibition, staurosporine is still very often used. It's a go-to in developing these kinds of assays. Now, its potency is quite high, and that manifests in a low nanomolar IC50. And don't worry, I'm going to explain how to measure that shortly. Okay, so how is it that we study kinases and inhibitors? So the tool that we use that we're really experts in are biochemical kinase activity assays. So what is an activity assay? Well, what that means is that we mix together the kinase of interest, the substrate, the inhibitor, all the buffers and cofactors, the manganese, magnesium, DTT, whatever it is that the kinase enzyme needs. And then we add ATP, and then we're off to the races. So if we want to detect the rate of this reaction, one way to do it is to look at the product, the phosphorylated product. And of course, we can do that with phosphospecific antibodies. But every kinase produces a different phosphorylated product. On the other hand, every kinase produces the same side product, ADP. And SignalChem has a long-standing collaboration with Promega, who produces a kit that you can add that produces a visible luminescent signal from the ADP produced by this reaction. You're going to see some pictures of that later on. The key to this kind of assay is that this is an in vitro assay in a test tube, where we're not worried about other things in the cell or the cell membrane or anything else. We're really able to probe directly the effect of the inhibitor on the kinase of interest. Now, this is not just binding. We're measuring activity. It's really important to point that out because those two are not the same thing. So let's say we have two inhibitors and we want to compare them to each other. We do that by measuring their IC50s. So the way that we measure an IC50 is by starting out with the kinase uninhibited. and measuring its activity, and then titrating in that inhibitor until we get to halfway, 50%, between fully active form and the fully inhibited form. So another name for IC50 is the half maximal inhibitory concentration. So one of the tricky things about IC50 is that we want it to be lower. The reason why is that that means we need less inhibitor or less drug to be effective. Now, what we're showing here are two different companies' kinases. We have ours and we have one of our competitors. Because ours are optimized to be more active, we get a better fit to a curve and a more precise measurement of IC50, as opposed to a less active kinase, which gives a less reliable readout. This might not be that important for this one example, but when you're comparing thousands of compounds, it's really important for you to have precise IC50 measurements. Okay, but so far we've just been talking about inhibitors. But a drug is not just an inhibitor. It also has lots of other properties. It has to be absorbed by the body. It has to be, it can't be excreted too fast. It has to be safe, and it has to be effective. So what's the first kinase inhibitor drug? It's imatinib, and it was released in 2001, developed by Novartis, based on one initial hit, which is this part of the scaffold here. And then medicinal chemistry was done to add these additional functional groups to improve its properties. It was developed to treat chronic myelogenous leukemia. But this oncologist here at Dana-Farber realized that it could also work for gastrointestinal stromal tumors. And he ran a clinical trial for it even before it was actually fully approved. So this happened in 1999. And of course, he became an oncologist because so many people he knew had had terrible experiences with chemotherapy. And to him, imatinib was a revelation. He called it the beginning of a new era of precision medicine. Like at the end of a James Bond movie, you cut the red wire to stop the bomb. But in this case, we stopped the signal with a pill and the cancer cells died. This was such a big deal. This was on the cover of Time magazine. You know, nowadays, I think we need to remember how difficult things used to be before kinase inhibitors. So this was just the first kinase inhibitor. Let's take a look at what happened to the development of kinase inhibitors between then and now. So Nature Reviews Drug Discovery in 2021 made this timeline that shows basically an exponential growth in kinase inhibitor drugs. Now, most of these drugs were developed for oncology purposes, but the ones here in blue are for non-oncology purposes, things like neurodegenerative disease and autoimmune disease. The pink entries here are therapeutic antibodies. So it's mostly small molecules and also some therapeutic antibodies mixed in. Each of these drugs that was released took a huge process to be developed. Let's take a look at what that process is and how SignalChem Biotech can help. To get small molecule drugs, we use a process called drug discovery. And an analogy for drug discovery that people often use is a funnel. And that's because we start with tens of thousands or millions of compounds and discard compounds that don't have the properties that we want. But what are those properties? The first step of drug discovery really is identifying and validating a target. And in this case, what we're talking about is a kinase whose misregulation leads to disease. Once you have that target, then you can use high throughput screening to look at these thousands of compounds. It's important to have good assays, both biochemical assays like the kind I've talked about, and also cell-based assays to assess the quality of the compounds in the stage 1 early drug discovery process. Now, at this point in the process, people don't want to get too attached to any one molecule. So they don't even call it a lead, they call it a hit when it has some good property towards the target. But what I don't like about this metaphor is that it looks like all the molecules just get dropped in at the top, but that's not really how it works. When a hit is found, it's sent back to the medicinal chemists who synthesize a whole bunch of analogs. And then those compounds get put through the same screening and biochemical assays again. It's an iterative process, developing molecules to be more and more in line with what is required to treat the disease. There isn't a hard and fast line between stage 1 and stage 2, but oftentimes stage 2 involves animal studies. And you can't really screen 10,000 compounds in mice. But when you have a smaller number, a short list of compounds, that's when you can do these kind of animal studies and see whether or not it's effective. And after all of that development, in which of course in stage 2, there's still more additional medicinal chemistry being done to optimize the properties, when you have a short list, those get sent, the best candidates get sent to the clinic and tried on humans. And still out of those, the majority of those also fail to be approved and become a drug. What we're going to be focusing on is the stage 1 drug discovery. So let's take a look at what one of these high throughput screens actually looks like. So this is a picture of a real high throughput screen provided to us by Hisham at Promega, who helped develop this glow assay that each of these dots is kinase activity. And the brighter the dot, the higher the kinase activity. So the typical methodology for high throughput screening is that we're looking at the kinase target that we've selected based on the target validation on the entire plate. Every well has that same kinase, and each member of your library goes into a different well. And when you see a dark spot where there isn't kinase activity, that's considered a hit. Now, later on in your drug discovery process, you might ask a different question. You might have a particular compound in mind, but you want to know how selective it is for your target. In that case, we do compound selectivity profiling, where we put the same compound in every well of the plate, but in each well, we put a different kinase. So let's take a look at what some results of that look like. So in this table, every row is a different kinase, and every column is a different small molecule drug or inhibitor. The blue is basically not inhibited. White is a little bit of inhibition, and then red is a really strong inhibition here. So what you can see is that some small molecule inhibitors are very specific for their kinase targets. For example, tofacitinib pretty much only inhibits JAK3. On the other hand, some other ones are a little bit more promiscuous. So, for example, gefitinib, it affects a few members of the TK1 family. And dasatinib is broadly inhibitory to the whole TK2 family, in addition to some members of the TK1 family. So this is really important information to know, because inhibiting an off-target kinase can lead to side effects, and that can really affect your ability to get through your clinical trials. So I'm at the halfway point. Before I pass it off to Jesper, I just want to summarize and say that kinases are a proven and growing class of drug discovery targets. If you want to do drug discovery on kinases, kinase activity assays are a really important part of that process. And if you want to get really good screening results, use high activity kinases like the kind we make at SignalChem Biotech. All right, Jesper, I'm going to pass the mic over to you and let you talk about it.
Well, thank you, Adam, for that very excellent introduction. So at this point in time, we've reached the halfway stage of the presentation, and we're going to be changing gears a little bit and transition over to the story of how SignalChem Life Sciences utilized the capabilities found within SignalChem Biotech to perform high throughput screens and kinase biochemical assays to investigate potential new targets against the receptor tyrosine kinase Axel, which has been implicated in many different kinds of cancer. So through this part of the talk, I will be using our drug discovery funnel over here as sort of a indicator of the progression as we go through the process itself. It's a very rough sort of a way marker. And I'm going to start with the first stage, which is the drug discovery part itself. And the first thing that we had to do here at SignalChem was to validate that our target, Axel, was actually a druggable target. Once we had identified that that was the case, we then started doing our initial drug library compound screens to identify the first couple of hits. And then we went on to do medicinal chemistry and optimize the leads based on specific characteristics for the drugs that we were interested in. The drugs that we wanted needed to be specific for the Axel kinase. It needed to be able to downregulate downstream targets of Axel itself and also inhibit cell proliferation. In addition to these characteristics, it also needed to have a good ADME and pharmacokinetic profile, which I'll talk about a little bit more in detail in a couple of slides. But we'll return to that aspect of things a little bit later. But before we go into the data analysis, I'm just going to quickly spend a second here describing what is Axel. What does it do? Well, Axel is a receptor tyrosine kinase from the TAM family, which also encompasses Tyro3 and MER. As a receptor tyrosine kinase, it is a transmembrane protein that has an extracellular domain which interacts with its ligand, and an intracellular domain where you have the kinase activity of the molecule itself. The way that Axel works is that upon binding of its ligand, Gas6, it causes dimerization of Axel, which leads to autophosphorylation of the kinase itself. Once the kinase has been autophosphorylated, it will start various signaling cascades, activating other downstream kinases, leading to changes in gene expression, which results in increased cell proliferation and cell survival signals. Important to note from this particular part of the slide is the autophosphorylation and also one of the downstream targets known as AKT, as I will be returning to those multiple times throughout the rest of the webinar. Now, since Axel is directly involved in regulating cell proliferation and cell survival, it is maybe not surprising that it's also very prominent in a variety of cancer subtypes. And for that reason is a very, very interesting target for the development of new drugs that are targeting the Axel kinase itself. Now, while it might be interesting as a target, that doesn't necessarily mean that it actually is a target. So the first thing that you need to do before going into a drug development process is to verify that, yes, the target that you have in mind will work for the development that you have envisioned. So to address the issue whether or not Axel is indeed a target for inhibition, the first thing that we did was we did an siRNA screen of Axel. And the idea behind this screen is if Axel is indeed involved in the signaling that leads to cell proliferation, then knocking down Axel should also lead to a reduction in the signaling that comes downstream of Axel, which has been linked to increased cell proliferation. So what you're seeing here is two Western blots. The one on the left is investigating how Axel responds to transfection with siRNA compared to a beta-actin loading control. And as you can see, increasing amounts of siRNA leads to reduction in Axel protein levels in these particular cell lines. Now, the other part of this graph shows what happens downstream of Axel as we reduce the amount of Axel on cell surface. So what we see here on the right is AKT total amounts. And on top, we have phosphorylated AKT or activated AKT. And as you can see, as we increase the dosages of our siRNA, we see a reduction in the amount of phosphorylated AKT, but not total AKT. This indicates that indeed, when we reduce Axel, we reduce the signaling that comes downstream, not the protein levels. So that would check one of the boxes for a potential drug candidate. Now, the next thing that we need to know is, well, Axel may be regulating all of these downstream signaling cascades, but does it also regulate cell division and cell proliferation? To test this, we did a thymidine assay where we incorporated radioactive thymidine into dividing cells. And the idea behind this assay is that as your cells are dividing, they will take up this radioactive thymidine, and you can measure the amount of radioactivity that's incorporated in your DNA in all of the cell lines, and then you can get a measure of how much cell division is happening in specific conditions. Then you add your drug, or in this particular case, siRNA. Later on, we will be using the drugs. And then you measure the amount of cell division compared to your control. So how much are you reducing cell proliferation as you are treating your cells in the various cases? In our exploratory study to see whether or not Axel was a good target, we inspected the cells with an increasing amount of Axel and investigated cell proliferation. And here you can see inhibition is up on the y-axis here, and you can see that the higher the concentration of Axel, the stronger of an inhibitory response was observed, indicating that yes, indeed, reducing Axel will result in a reduction of cell proliferation. Now, importantly, it was not a transient effect that we observed. So we did the measurements at 24, 48, and 72 hours post-transfection to see, is this a prolonged response, or is it something that just happens transiently as we introduce the siRNA? And importantly, we can see that the inhibition is sustained for a duration of time, which is also important for a potential druggable target, that you see long-term effects upon the treatment. So at this point in time, we have validated that Axel indeed is a target, and we can move on to our drug screening and lead compound optimization. So what we did at this point is we started out with a library of compounds and did the initial screen, identifying various hits. And those hits were then modified using medicinal chemistry. So these modifications is anything from extending chain lengths or shortening chain lengths, changing functional groups, including more double bonds, removing double bonds. So any kind of chemistry that you can kind of envision happens as you're creating these analogs from your initial hits. And then you undergo another round of screening, and this is an iterative process until you get compounds that have the properties that you want from your drug screen. So at the end of the screening, you would end up with a table that looks something like this. So here we have a bunch of numbers of various compounds, and it's kind of a lot of information to take in. So what are the parameters for us to select which of these many compounds that we want to continue on with in our drug screen? Well, first of all, our compounds needs to be specific for Axel. And what you're seeing here is the IC50s from the 3 TAM members. So you have Axel on the left, you have Tyro3, and you have Merck. And the desirable properties here would be to have a low IC50 for Axel, and not as much for the other members of this family of receptor tyrosine kinase. The second property that we're looking for is its ability to inhibit downstream activation of Axel. So here we're looking at the phosphorylation of AKT as the readout for our ability to inhibit this signal transduction pathway. And again, we're looking at a low IC50 as an ideal scenario. The last thing that's important here is looking at the ability to also block cell proliferation. So we want to have the whole package where we get both knockdown of the signaling pathway and also blocking cell proliferation. And again, we're looking at a low IC50 for all of these compounds. The last thing that we also do for the compounds at this stage is we do an ADME and pharmacokinetic analysis, where we look at the behavior of the compounds in cell tissue and in animals as well. And when I say look at the behavior, we're looking at things like, is the drug converted to metabolism? Can your cells actually take up the drug? Does it end out in the target tissues as wanted? How much of a percentage of the initial compound is actually arriving at the target, and those kind of parameters. So we're basically investigating what will happen once a patient would take this drug. Does it get to the right locations, or is it just metabolized into something and doesn't really survive the environment of an organism? So when taking all of these four parameters into consideration, we identified SLC-391 as having desirable properties for subsequent analysis. Sorry, need a little bit of water. Okay. So the next step that you then take is once you have your compound, you go in and do a kinome selectivity profiling study. You want to make sure that your compound of interest is highly specific towards the kinases that you actually want to target. So here we have an example of a kinome selectivity profiling assay where the compound itself had hits in various branches of the kinome tree, as you can see here. Now, that is not desirable when it comes to an actual drug, because you might have a very large number of unintended side effects and ramifications that you're not intending if you start inhibiting kinases all over the board. So you want something that looks much more like this. Now, it's important to note that the first of these studies that we do here at SignalChem Biotech is all biochemical assays, meaning that, as Adam described, we are adding in the kinase, we are adding in the inhibitor, we are adding in ATP and substrates. So it is very much so an artificial setting from a cell perspective. So once we have done the initial screening of our compounds and identified that they don't have a ton of side targets, then we can take them to the next step, which is a cell-based assay, where we use a cell line known as BAF3 to better illustrate how the drug behaves when exposed to a cell environment. So these BAF3 cells are a cell line that have been created so that they are dependent on the presence of interleukin-3 in the growth media itself. So basically, in order to keep these guys alive, you have to add in the interleukin-3, or they will die. What you do with these cell lines is that you transfect them with receptor tyrosine kinases, one for each of the cell lines, and then you allow that receptor tyrosine kinase to take over the signaling that maintains cell viability. So you're basically doing a transfection where now the receptor tyrosine kinase takes over the job of interleukin-3, so you no longer need interleukin-3 in the cell media. Once you have created these stable cell line and verified that there's only one kinase for each cell lines, you now can add in your drugs. And if the drug inhibits your kinase, then you will induce apoptosis, and you can measure the amount of ATP that's left over in the various wells when doing this kind of assay using cell titer glow. So what we do is we make a large panel of these transfected cell lines, and now we test them using our drug of choice. So when we did that for SLC-391, we saw that it's specifically inhibited Axel and MER from the TAM family, and only had a couple of additional hits in the form of VEGFR and TGFR. Now, this is a very clean profile, and one thing to note here is that we do add a lot of drugs when we run this particular assay. 100 nanomolars worth of drug for each of the lanes, which is much higher than the IC50s. This is a profile that is promising in terms of taking it on to the next step and then doing more analysis of this compound. So what is the next step? Well, the next step is to look at various cell lineages to see whether or not your compound actually inhibits the downstream activation of Axel inside of these tissue culture cell lines. And what we did here is we tested two different cell line types. One is a human leukemia monocytic cell line, which is known as THP1 cells. And what we did here is we added an increasing amount of SLC391 in the presence of the activator Gas6. So what you're seeing here in the Western blot is in the first lane, we have our control lane. We have no Gas6 and we have no SLC added. And we are looking at the relative levels of Axel and AKT and the phosphorylation state of both of them. We then introduce Gas6 to show that we can stimulate autophosphorylation of Axel and also downstream activation of AKT. Now, important to note is that as we increase the amounts of SLC391, we see a reduction in the phosphorylation of both Axel and its downstream target in AKT, showing that SLC391 in the cell line has the desired effect. Next, we moved on to do the similar kind of study using another cell line, which is an adenocarcinoma human alveolar basal epithelial cell line, also known as A549, where we did the exact same study. And I'm not going to belabor this too much. We saw the exact same results, whereas when we increased the amounts of SLC391, we saw a reduction in both phosphorylation of Axel and also AKT. So we can see that the cell lines are inhibited when we add our Axel inhibitor. At this stage, we are ready to move on to the second stage of our drug development funnel here and move into the preclinical development, where we look at the ability of SLC391 to impact both leukemia and adenocarcinoma models in mice. And what we're looking for here in terms of positive results is that the treatment should reduce cancer phenotypes. It should perform well compared to other known anti-cancer drugs, and it should expand the lifespans of our mice. The first of our experiments that we did was a mouse experiment where we irradiated mice with cesium and destroyed their bone marrow. And then we injected them with a stable cell line that's expressing GFP and luciferase. And importantly, this is a leukemia cell line. So it basically behaves as leukemia upon the injection into the mice. Since it's stably expressing GFP, we can measure the leukemia in the mouse by putting it on a scanner and detecting where is the fluorescence. We then treat the mice with the SLC391 and a known BCL2 inhibitor, venetoclax, and we then look at what's happening to the leukemia and the survival of the mice. So looking at the data here, we can see that in our untreated mice, the leukemia is found pretty much throughout the mouse, but treating them with SLC391 significantly reduces the amount of leukemia signal that we see in the mice. Also compared to the known BCL2 inhibitor, venetoclax, SLC391 performs even better than this one. Now, important to note is that a combinatorial treatment between SLC391 and venetoclax leads to an even better outcome, where you can see there's almost a complete absence of leukemia in 2 out of the 3 mice that are indicated here. This is also reflected in our survivorship curves, where our combinatorial treated mice far outlive the vector alone control mice, or vehicle alone control mice. So this indicates that SLC391 works very well against leukemia. What about solid-state tumors? To test this, we xenografted mice with a non-small cell lung cancer cell line, and we looked at the tumor growth as a function of time. And as you can see in the vehicle controls, there's an exponential growth of our tumors. Whereas if treating them with SLC391 resulted in a reduction of 42% after 28 days, showing that yes, SLC391 does treat the solid state tumors. This led us to doing yet another combinatorial study where we looked at the function of SLC391 combined with another drug. In this particular case, an anti-PD-1 drug. So PD-1 is a receptor that's found on T cells that's involved in these immune checkpoint progression. So if this receptor binds, then the T cell will not kill your cancer cells. But if you have the antibody, then the receptor cannot bind, and you can now kill your cancer cells. So what I'm going to draw the attention to very quickly on this particular graph is that comparing the vehicle alone to our combinatorial study, we can see that the combinatorial study far outperforms each of the individual studies and also leads to much better outcome than the vehicle alone. So these combinatorial treatments are very, very effective. This is also reflected again with a survivorship curve, where our combinatorial treatment between SLC391 and anti-PD-1 leads to by far the best outcomes for the mice. Now, this is very, very promising, and that led us to move on into clinical trials, going into a partnership with Merck and using their anti-PD-1 drug, Keytruda, as part of these clinical trials. And as of May 31st, 2023, we entered phase 1b-2 of these clinical trials, which puts us somewhere at this particular juncture in our drug development funnel. So in conclusion, we have shown that kinases are very important regulators of cellular processes, and that they represent a very large target for additional drug development. And we have shown how SignalChem can help you doing kinase screening and profiling as part of the drug development journey. As an example of this, SLC391 has been shown to be a very potent inhibitor of Axel signaling, and it has shown to have anti-cancerous properties in mice and is currently undergoing clinical trials. So at this point in time, I want to thank all the people who did the work and the people who supported us putting this presentation together. I also want to thank Biological for giving us the chance to present here today. And at this point in time, I'm happy to take any questions, as long as they're not related to the clinical trials, as I am not at liberty to really discuss those in particular detail.
Thank you, Dr. Johansson. That was an excellent lecture. Thanks, of course, to you and Dr. Barlov. Really, really thorough all the way through from the initial introduction of Axel to the small molecule drug that is now, I believe, in clinical trials. Congrats to y'all on this wonderful work. It's very exciting. We're running out of time, so we'll jump right into the questions from our audience. First question is from Susan Paprika, if I pronounced that correctly. Forgive me if I didn't, Susan. And her question is, earlier in your talk, I believe, Dr. Johansson, you had given an introduction on the downstream effects of AKT and its inhibition, of Axel and its inhibition. And Susan asked, why did you look at phosphorylated AKT instead of phosphorylated Axel in your functional assay?
Well, I mean, what answer to that, thank you for the question, is sorry, I'll get closer. Can you hear me now?
Yeah, that's good.
First of all, thank you for the question. And we looked at both. We looked at the autophosphorylation of Axel as part of the analysis to see whether or not Axel actually initiated the process. And we also looked at AKT downstream to verify that there was a signal going from the phosphorylated Axel down through the signal.
Okay, great, sounds good. And Susan has a couple other questions. I'll get to her last one here. Would you expect the off-target activity against FLT3 to play a role in the cytotoxicity of the BAF3 cell line?
Oh, that is a very, very good question. And I will. Is it possible to return by email, because I need to think about this for a second? So,
yeah, we'll just have you reach directly out to Susan if that's okay. I believe we have.
Yeah, Yeah, I'll need to ponder this a little bit, and I know we're running a bit short on time, so
Yeah, no worries. I did think that was a good question. So,
oh, it's an excellent question.
all right, the next question is from Ike Uger Marion. Please forgive me again if I mispronounced your name. This is a great question. Did you also test mutated kinases like those that have single point mutations that may confer resistance to a drug, for example?
We can absolutely do that. We have a large and ever-growing number of kinases that have some of those resistance markers in them, so that can absolutely be part of a screen that's ongoing, where you look at the inhibition profiles and you look at the interaction between those mutants and confirmed drug resistance kinases.
Great, great. And let me just ask a follow-up question to that. And if you don't mind, just speak a little louder into the microphone.
Oh, sorry.
You may have said this and I just missed it, but like how many kinases do you have in your assays total right now?
So we currently, well, so the short answer to that is it depends. I mean, often we will run a panel with 93 kinases and run them as we go through a profiling run. Now, it depends a little on what you might want to investigate. So if you have specific families in mind, and if you have specific interactions that you're interested in, you can modify these screens. Typically, 93 kinase screen is what we do. But obviously, sorry, obviously you can modify in any way that is needed. So wonderful kinome screen that is.
Okay, Dr. Johansson, I think we're having some audio problems. But let's jump to another question. Maybe we can try once again with some. I want to try to get to as many as possible if we can. So Neil Anand is asking, what was the model, tumor line, and immune system used for the anti-PD-1 combo study?
Yeah, I don't answer that. Is that what they're looking for? Okay. Let's go back to that slide.
Yeah, and please try to speak directly into the microphone. I don't know why we're all of a sudden having problems.
It was a non-small cell lung cancer line. Did you get that? Can you hear that?
Yeah, we can see it.
Okay. No? Still can't hear me?
Yeah, I can hear you. Sounds good.
Okay, good. Yeah, what other questions have you guys got?
Let's see. So second question from Neil. What level of Axel inhibition was being achieved by the SLC 391 small molecule at the dose level tested in that model? I'm not sure if you understand that question.
Yeah, so it's right here on the slide, actually. So it's 50 milligrams per kilogram is the concentration that was used in this particular study. And for, how that relates to the IC50, the IC50 is also in the low nanomolar, so quite potent as well.
Great, thank you for that. We'll get to one more question. This is a good question, and we'll wrap it up. This question is from, Afadinho Michael. Afadinho Michael is asking, first of all, Afadinho said it's an excellent presentation, and then is asking if you worked on actually identifying the active site of the Axel kinase, and if there are any other like allosteric sites.
Yeah, it's a good question. But there's a lot of details of this project that we can't disclose. But what I could say is that in other drug development programs, there are definitely attempts to make those kinds of inhibitors. There's sort of different kinds of kinase inhibitors, and what we've been focusing on here are the largest class of kinase inhibitors, which actually go directly into the ATP binding site and block ATP binding. So those are what we focused on in this talk, but certainly there's programs trying to look for all of the kinds of kinase inhibitors out there.
Sounds great. We actually have time for one more quick comment from you. Shabir Hussein is commenting that many patients actually don't respond well to anti-Axel treatment, or they actually acquire quick resistance to these agents. What are your thoughts on that?
I think that the business people behind the partnership with Merck have looked at all of these kinds of eventualities, and they've made the decision to go forward with this molecule. But certainly acquired resistance through mutations is a problem. And when those mutations occur, we will try and figure out what they are and add them to our product list so that you can try and inhibit them.
Sounds great. Thank you, Dr. Barlev. And with that, we'll wrap things up. Let me just thank everybody really quickly. First of all, I'd like to thank the attendees. We had a huge turnout today. No surprise there, because it's a very interesting topic with our new partner. We had folks attending from France, the UAE, Ethiopia, Germany, Italy, India, Algeria, Nigeria, Lebanon, of course, the United States, Iran, Mali, and even St. Kitts in the Caribbean. Wish I was there. So we appreciate y'all attending. Certainly speakers, we want to thank you, Dr. Barlev, Dr. Johansson. Excellent work. It's an honor to have you today. It's also an honor to be new partners with y'all and the rest of the team at SignalChem. We are really looking forward to working very closely with y'all for many years to come. And then finally, I will thank my colleague and good friend, Max Bleckman, who actually executed the technical aspects of this webinar and did it once again flawlessly. Max, we appreciate your efforts today. And with that, we will sign off and meeting adjourned.
Thanks, Rob.
Bye-bye.